Alright, welcome to your video for lecture 7b and 8. So before we get started, just wanted to say hi to everybody again. So at this point, you should be finished the material for midterm 1. So midterm 1 material goes from lecture 1b to lecture 7a. And so most of you are probably watching this having finished midterm 1 already, and so we'll get that marked as soon as possible, and I'll tell you more about kind of how to learn from midterm 1 in a little while. So I'll post a separate video for you guys for that. So for now, I'm just going to move on to the material that's from here on out going to be covered in midterm 2 and for the cumulative final exam for now. But before getting into the new lecture material, I just wanted to talk a little bit about study habits and what to do at this point, since you're probably quite in the thick of the beginning of midterm season now, so you're gonna have midterms probably every week or maybe several per week from now on. And so now would be a good time to really try to set aside and cut back on the number of distractions that you have affecting your study time. And so some tips on how to do that. So if you're one that gets sucked into social media quite a bit, one tip would be to just put on some sort of away message for your social media. Just post something saying that I'm going to be busy until the end of the semester, so don't expect me to comment on things. And then from then on, it seems kind of extreme, but it would work, is get a friend or a roommate or a parent to change your password for Instagram, Facebook, even your Xbox access key and your Netflix access as well, just so that you remove those temptations. And you can still take breaks and watch a bit of TV here and there, but for whatever tends to suck away your time away from studying and make you regret kind of wasting time, now's a good time to have someone change those passwords, just so you don't get distracted by these things, so you can focus on your studying, get it done, and then you can fully enjoy these things again after midterms are over, and you can freely enjoy these distractions once again. Okay, just a few tips there. And so with that, let's move on to your new lecture material. All right, so lecture 7b and 8 is about looking at gene interactions, particularly looking at complementation tests and biosynthetic pathways. So in the previous lecture 7a, you looked at kind of interactions between different mutant alleles, but only kind of one gene at a time. So for example, when we're looking at interactions between codominant alleles, right, we're still looking at different alleles for, say, the flower color gene, or even when we have kind of an allelic series where there are several alleles that kind of interact in a dominant recessive way across a series of kind of decreasing dominance, we're still just looking at multiple kind of different mutations, different mutant alleles of a single gene. So for this lecture, we're going to, and for the ones that come after this as well, we're going to start looking at different interactions between more than one gene. So the first thing we're going to do for this lecture is kind of help you figure out whether you're looking at a situation where different mutations are affecting the same gene, kind of like we see here, or whether we're actually looking at a situation where more than one gene is affected by the mutations you're studying. All right, so we'll start with that. And then we're going to kind of look at different gene interactions based on genes that affect a particular biosynthetic pathway. We do that by studying oxytrophic mutants in order to figure out the steps of a biosynthetic pathway. So you may not understand what any of that means, but you'll know by the end of this lecture. All right, so that's just a bit of outline of what we're going to cover today. So the first thing we want to figure out is, if we're studying kind of more than one mutant trait or mutant strain, one thing we want to know is whether we're looking at mutations that are affecting just one gene or more than one gene. All right, so here's a bit of like a case study situation where I'm going to kind of walk you through what a scientist might come across and get you to think about what decisions you'd make and your interpretations of the results, and get you basically thinking like a scientist. Okay, so let's say that you're doing like an honors thesis or doing co-op, and while you were working with some kind of lab-reared housefly colonies, you discovered that there were some flies that had no wings. And it seems to be a genetic kind of trait. It's not like they lost them in a fight or something like that. All right. And you, with a bit more studying, found out that wingless trait is produced by a recessive allele. So when you have a fly that's homozygous for this recessive mutant allele, then the flies have no wings. So since you're really interested in this and your principal investigator supervisor is also interested in it, you took some time to and resources to isolate a pure breeding population of these wingless flies, such that both the males and females are all wingless, and when they have offspring, all of them are wingless as well. So you now have a pure breeding mutant wingless population. And since you discovered it, your prof said, okay, you can go ahead and name it. So being a proud Canadian, maybe you named it the Canadian mutant strain. Okay, these are the Canadian wingless mutant strain. Okay, so when I use the word strain here, I'm talking about a population of flies, right, males and females all mixed together, but it's pure breeding for this particular mutation, meaning that it's in terms of the mutant allele, it's homozygous for that mutant allele, which is why all the offspring has the same is homozygous for the same mutant allele, and that's why all of the offspring are also wingless. Because that's what I mean by strain is a population that's homozygous for this particular interesting mutant trait that we observed. Okay, it is important that it's a recessive mutant allele. Keep that in mind. Okay, so let's say that you've done this work and you discovered this strain. You're really proud, and your parents are proud that we now have this new mutant allele that's named that you've named. Now, let's say that on the news later on, you see that there's another researcher in Harvard that also found a brand new recessive allele that also produces a wingless phenotype. All right. So this person, being a proud American, maybe calls it the USA mutant strain. So she has a population that is also pure breeding for this wingless mutation of hers. All right. So since you're kind of interested in pursuing this kind of mutation and the genes involved in producing wings further, you email this researcher and ask her to have some of her flies. So you request them from her. And we actually very freely share materials and information between labs a lot of the time. So if you ask that lab to send you some flies, she'll do that. She'll put it in a package, put the right shipping labels on it. It'll take a while to get through customs, but hopefully if all the shipping stuff is done correctly, you'll get some live flies delivered to your lab before they die from, being stuck in storage for too long. All right. So once you now have in your lab your own mutant strain of wingless flies and the Harvard kind of USA mutant strain of wingless flies, the first thing you're going to want to do is try interbreeding them, try crossing them together to see what happens. So let's take a look at the results of this cross, right? So we take some flies from our Canadian mutant strain and mate them with the USA mutant strain, and let's predict what the offspring will look like, right? So if we hypothesize, if we assume, for the sake of this prediction, that different genes were affected in the mutation that caused winglessness in our strain compared to a different gene being affected in the USA mutant strain. Okay, so if you assume that different genes were affected by these within these two mutant strains, then what would you expect to see when you cross these two mutant strains together? All right, so this is a kind of question for you to try answering in your participation quiz. So you can pause the video and make your choice and really think about this one. It takes quite a bit of thought to figure this one out, but see what you can figure out. So go ahead. All right, welcome back. So the correct answer is A. All right, so if the gene that's affected by the mutation in your strain is different from the gene that's affected in this other strain, you would expect all of the offspring to be wild type, to be fully normal, to have wings again, even though neither of the parents had any wings. All right, so why would you expect this? So you didn't get that question correct. That's totally fine. That's a very difficult question, but if you got it right, great job. So let's talk about why we would expect that result. So if two different genes were mutated between these two different mutant strains, we would expect all normal wild type offspring. So let's take a look at it in terms of the genotypes carried by each of these strains. All right, so let's focus on our own strain first. So let's say that there's two different genes being affected, right? So in our Canadian mutant strain, let's assume that there's a gene, we'll call it the CAN gene for Canada, okay? And we are homozygous, since it's a pure breeding strain that we have of this mutation, then we know that it's homozygous for the mutant allele. So notice I don't have a little plus here, right? Which is my way of indicating that these are not wild type alleles. These are the messed up mutant alleles, and we're homozygous for that mutant allele. Okay, you can also see it in this picture here that the Canadian gene has some sort of mutation happening in this part of the gene, which is why it doesn't work, which is why we have no wings. Okay, for the American mutant strain, you would assume that, okay, they have a different gene, say the USA gene, that is homozygous mutant 4. So there's a completely different gene with a unique mutation there that shuts it down and makes it not work, such that it's also not able to make wings, but for a different reason. All right. But notice when you look closely at the genotype, well, since these are the same species, right? Each species has 2 copies of the Canadian gene, 2 copies of the USA gene, right? Our strain is homozygous for these mutant alleles of the Canadian gene, but presumably its USA gene is fully normal, right? That our winglessness is caused by problems with the Canadian gene. For the USA gene in our strain, it should be still homozygous wild type, right? We didn't collect any mutations there. For the USA mutant strains, the other way around, right? It's homozygous mutant for this particular mutant allele for the USA gene, but presumably its Canadian gene was not affected by any mutations at all. That's still fully wild type as well. All right. So when we cross these together, right, we're going to look at their offspring, right? The offspring are going to receive 1 copy of the Canadian gene from our strain, which is going to be mutant, and 1 copy of the USA gene from our strain, which is going to be wild type, because that's all that our parent kind of Canadian mutant strain has, is messed up versions of the Canadian gene and normal versions of the USA genes. That's what it gets, the offspring gets from this parent. From the other parent, we get a messed up copy of the USA gene, because that's all that this strain is carrying. But it's also carrying only wild type copies of the Canadian gene, so we get a good copy there as well, right? So overall, the genotype for these crossed offspring, these kind of hybrids, you could say, of the Canadian and USA mutant strains, will be heterozygous for both genes. And since the wild type allele is dominant, right, then we have wild type phenotype for Canadian gene, wild type phenotype for USA gene. That's why the offspring are all going to have wings, right? So since the offspring have a wild type dominant allele for the Canadian gene and a wild type dominant allele for the USA gene, then they should still be able to make normal wings. Okay, so that's why if a different gene is affected within each of these strains, all of the offspring should have normal wings. Okay, so this is an example of a complementation test. Okay, so this is a particular kind of cross that we do when we have 2 different mutant strains to see whether the mutations in one strain affects the same gene as the gene that's affected in the other strain. Okay, or are there 2 different genes being affected? So one gene affected here and a different gene affected with the other strain. All right, so the way that we do a complementation test is what we did here, right? We take 2 pure breeding strains that are pure breeding for its own recessive mutant allele, all right, and you cross them together, you mate them together and look at the offspring. You basically look at offspring to see whether all of them are wild type. If all of the offspring are wild type, as we saw in this example, then we say that complementation occurred, and from seeing complementation occur, seeing that all the offspring end up being wild type, we would conclude that the mutations seen in one strain affect a different gene from the mutations found in the other strain, that there's 2 different genes being affected by mutations in these 2 different strains. But if you find that the offspring are not all wild type, then we'd say that, oh, it's failure to complement then, right? Since we didn't see all wild type alleles, we say we failed to complement, and what we would conclude is that the mutations affect the same gene, okay? So if that's a little confusing to you, don't worry about it. We're going to look at each of these 2 situations much more closely in a minute. But for now, I just want to help you understand the terminology here. So when we say that one strain of flies is complementing the other strain, right? Notice complement with an E, not complement with an I, right? So when I say that one fly strain is complementing the other one, I'm not saying that one fly goes to the other one and says, hey, sexy, nice thorax. It's not giving them a compliment. When we say complement with an E, we're talking about something like, complementary base pairing, right? So if you think about how G and C, guanine and cytosine bases, are complementary to each other, how they form a complementary base pair, it's because they're not the same, right? G's and C's are not the same at all, but one's bigger and one's smaller, and their kind of slightly positive and negative portions allow it to base pair nicely. So by complementing, we mean that they're different, but they come together to complete each other, right? So the bigger part matches the smaller part, the positive parts match the non-positive parts, and therefore they're meant to join together. Okay, so to give you another example of what I mean by complementation. So let's say that for some reason I really don't like buying groceries. I'm actually not a big fan of buying groceries, right? So let's say that there might be some gene in me that's mutated such that I just don't have the enjoy buy groceries gene, but I do have the enjoy cooking gene. right? So if my goal is to have some home cooked meals, I'm going to fail at that because as much as I like to take raw food and cook it, if I don't buy groceries, I have nothing to cook at home. Okay. Now let's say my wife doesn't have my weird genetic flaw and she's totally fine with buying groceries, but she has another flaw that runs in her family where they don't like to cook. Okay, so in that case, again, we're not going to have any home cooked meals because as much as she likes to buy groceries, she doesn't like to cook, right? Now, let's predict what's going to happen with our offspring then. So let's say Katie happens to get a good copy of each of these genes, right? So I might be homozygous recessive for this mutant don't like to buy groceries allele. She might be homozygous recessive for this don't like to cook allele, right? But we each are going to give a normal dominant wild type copy of each gene to Katie. All right. So as a result, she should be able to buy groceries thanks to getting a normal wild type gene from or normal wild type allele from mom for that. She should be able to cook that raw food into a home cooked meal because she got the normal wild type copy of that gene from me, and therefore will hopefully have some good home cooked meals when Katie gets a little bit older. Okay, it's kind of a simplified example, but that kind of shows you what I mean by complementation in that, you know, when I do a complementation test of my inability to make home cooked meals and my wife's inability to make home cooked meals, right? We see complementation. We see that our offspring are all able to make home cooked meals, not because we're the same, but because what I'm missing, my wife has. What she's missing, I have, right? And that's why we complement each other. Okay, so complementation means that we're different in a way that matches up nicely, such that our offspring will all have what they need to be wild type to finish the reactions and produce what a normal person should be able to do. Okay, so to apply that to our wingless mutations, right? You can imagine that in order to produce wings that allow a fly to fly, that requires a lot of steps. A lot of steps are essential to making wings. So you can imagine that for our Canadian mutant strain, we have a recessive null mutation, meaning a mutation that is so messed up that the gene no longer produces a viable, useful product, right? So we have a null mutation in a gene that's needed, like maybe it no longer makes viable enzyme for catalyzing a particular reaction, and that's a necessary step to making wings, right? So since we're homozygous for messed up versions of the gene needed for this step, then we can't make the step, and therefore our strain can no longer make wings, right? The USA mutant strain, since a different gene is affected, it's got a null mutation in a different gene, that means a different step along the series of steps must be affected, and also it has no wings, but it's having no wings for different reasons because different steps are affected and different genes are affected, all right? But because different genes are affected, then when you look at their offspring, the offspring are able to do every single step, right? They get a good copy of the gene needed to do this step, so they can do this early step. So they got the good copy from the USA mutant strain. And then for this other step, they weren't able to make it in the USA mutant strain, but since it's heterozygous for that gene and it's got a good copy from our strain, then they can do this step as well. So since what's mutated, the gene mutated in this gene is complemented by the wild type allele, the gene that's affected by this mutant allele that is homozygous for in this strain is complemented by the wild type here, then the offspring will show complementation and therefore be able to make wings. Okay, so that's hopefully a fairly clear example of what I mean by complementation and why it is that when you take two strains with different genes being affected, all of the offspring should be wild type. All right, so if that's confusing at this point, now would be a good time to go back and review what we've covered so far. Okay, but if that makes sense and it's starting to become clear, let's move on to the next step. Okay, so let's change our assumption then. So we made this prediction of all wild type offspring based on the assumption that different genes are affected within our two different strains. All right, so let's change the assumption now. So let's make some predictions based on the assumption that the mutation in our strain affects the same gene as the mutation in the USA strain. All right, so if the mutations affect the same gene instead of two different genes, what would you expect to see in the offspring? All right, so pause the video and make your choice. All right, welcome back. I'm going to give you a little more explanation. If you want to change your answer, you can after that. All right, so let's try kind of mapping out what the the chromosomes or genes might look like for our two strains if we assume that the same gene is being affected. All right, so let's say that there's a gene called the M gene. All right, so for our strain, we'll be homozygous for a particular mutated version of the M gene, right? So it's maybe there's some sort of deletion or some change in the base sequence here, such that this gene no longer produces a useful product, and that's why you can't make any wings, right? So we're homozygous for that particular mutation, which we call the Canadian allele, Canadian mutant allele for this gene. So if we look at the USA gene, notice now that we're not looking at a different gene, we're looking at a different mutant version of the exact same M gene. It's just that the mutation might have happened in a different part of the gene, but regardless of that, this is also a null mutation. It also does not produce a working product, and therefore we also have no wings. All right, but because it's the same gene that's mutated in both of these strains, when you then cross them together, what you'll find in the resulting offspring is that they get one messed up copy of the gene, so the Canadian mutant version of the M gene, and then the USA mutant version of the same gene. So we have one mutant allele mixed with a different mutant allele, and overall we'll have offspring that are all wingless because it doesn't have any working copies of the M gene. Okay, sorry, I kind of gave away the answer there, right? So notice the result is the opposite, right? When it's the same gene being affected by mutations for both of these strains, then neither strain has any working wild-type copies of the allele to give to their offspring. So as a result, the offspring it's heterozygous. It's got two kind of different mutant alleles, two different versions of this gene, but no wild-type copy, and therefore what we see is mutant phenotype. Okay, so overall, if both mutant strains have mutations in the same gene, then neither parent has a wild-type copy of the gene to give. So notice we don't have M plus anywhere from either parent, and therefore not in the offspring as well, and therefore the offspring doesn't have a wild-type allele to allow it to make wings. All right, if we want to show it in terms of these kinds of developmental pathways needed to make wings, right? Our Canadian strain is homozygous mutant for a gene needed for the first step, and the USA strain, same thing, homozygous mutant for, in a different way, but for the same gene, therefore affecting the same step, and so it's wingless kind of for the same reason. So our offspring are still going to get two bad copies of this gene, two different bad copies, and this also can't do the step and therefore cannot make wings as well. Okay, so hopefully that makes things a bit more clear in terms of the two different situations. So when you cross two different pure breeding recessive mutant strains together, either you'll see complementation and all of the offspring will be wild type and you'll know that two genes are affected, or you'll find that all the offspring are mutant, and that tells you that the two mutant strains have mutant alleles for the same gene. Okay. All right. So with that in mind, let's take a look at kind of one more set of, or let's broaden the picture a little bit. Okay. So I'm going to show you a bunch of different mutant strains that we're crossing together, pictures of them as well as genotypes and phenotypic descriptions. And I want you to think about whether these pairs of mutant strains will exhibit complementation whenever they're crossed. Okay, so for example, if we cross a wingless, our Canadian wingless strain with the USA wingless strain, considering what these genotypes tell us, would we always see complementation, right? Or if we're crossing a purple-eyed, so we're looking at a different trait now, looking at eye color. If we have a purple-eyed mutant and a white-eyed mutant, so different mutant phenotypes and alleles for the same trait, whether we would always see complementation. So if we did this, would we always see wild-type offspring when we cross them? And then we have these two very different traits. So we have purple eyes for one mutant strain and vestigial or kind of shrunken looking wings for another strain. If we made those together, would we always see wild-type offspring or see complementation? And another option is if we take a Canadian wingless strain and made it with a new strain that we got from Tokyo that's also wingless, would we always see complementation, right? So those are your four options of thinking yes or no for each of those, and then kind of choose your answer based on this, right? So if you choose B, then you're saying that, okay, we're always going to see complementation in 1 and 2, but not in 3 and 4 is what B is saying. Okay, so with that explanation, go ahead and pause the video and make your choice. All right, welcome back. So, of these four options, right, one that I would say would not always give us complementation is number four, right? Because we know nothing about the Tokyo wingless strain at this point. So it could be that the gene that got mutated in this strain to cause winglessness is the same gene as the one that got mutated in our strain, right? If that's the case, then it could fail to complement, right? Or it could be a different gene and it will complement, but we have no idea. We know nothing about the Tokyo strain for now. So that we cannot be sure that it will exhibit complementation, but the other three will, right? So answer is C, because these three kind of complementation tests will exhibit complementation. Now let's talk about why and why I wanted to point this out. So this first one was the example we talked about before. So we have two different mutant strains with the exact same phenotype, wingless and wingless, right? But from the genotypes, we can see that the gene that's mutated, that's homozygous mutant for, is for one strain is different from the gene that's homozygous mutant for the other strain, right? So if two different genes are affected, then we should see complementation. All right. So in this next one here, we see two different mutant strains again for something else, for eye color, but notice that they don't have the same phenotype. It doesn't have to be wingless and wingless. It could be different mutants. Like normally they have red eyes. This one has a purple eyes. This strain has white eyes, but we can still see complementation even if they don't have the same phenotype, because what's causing the different eye color in this gene, in this strain, is different from the gene that's mutated and causing white eyes in this other strain. All right. Also, you can even look at, mutant strains that have completely different mutant traits, right? One affecting eyes, one affecting wings, right? It's kind of obvious in this case that it's probably two different genes affected and that you will see complementation. But the point of this is to show you that you don't only look for you don't have to only look for complementation between strains that are mutant in the same way or even for the same trait, right? As long as for any complementation test, as long as the gene that's affected in one strain is different from the gene that's affected in the other strain, then you will see complementation. You will see all wild type offspring. Okay, so hopefully that clarifies what a complementation test is used for and how to interpret the results. Okay, so we're only asking, is it the same gene or different genes affected? And if it's different genes affected, we will see complementation. We will see all wild type offspring from this cross. Okay, so let's move into a slightly different twist to the situation. So we can also do complementation tests for recessive lethal mutations. Remember that these are mutant alleles that if you have two copies of that mutant allele, then you don't survive, right? You either have miscarriage or you're an egg that doesn't hatch, or you're an immature individual that never survives to sexual maturity. Okay, so that's what we mean by lethal, that you die before you have a chance to reproduce. Okay, so as much as this seems like an exception that maybe it doesn't happen very often, you might not study it, but this is actually something you'd actually want to study, because if there's a if you discover a recessive lethal mutation, that's worth studying, because any gene for which you can find a recessive lethal mutation basically tells you that that gene, the wild type version of that gene, is essential to the organism's survival or development or growth in some way, which is why when you don't have a working copy of that gene, you die. Okay, so basically lethal mutations are worth studying because those genes are really important for your survival. Okay, so if you were to discover a brand new recessive lethal mutation, one of the things you'd really want to do early on is see whether you've discovered a lethal mutation in a brand new gene, and basically you can say that, oh, this gene is super important because I found a brand new mutant allele that affects survival, or it could just be that you found a different way to mess up an important gene that someone else has already discovered. So essentially, you want to do complementation tests to find out whether what you've discovered is a new important gene or one that someone else has already discovered. Okay, so you want to do complementation tests to answer that question. But then the problem is, remember that for a complementation test, what we start with is pure breeding mutant strains, right? And we can't make pure breeding strains of recessive lethal mutations because they would die. They wouldn't survive long enough to reproduce to give you a population that can actually survive and reproduce again, right? So all homozygotes or hemizygotes, if it's a sex-linked gene and there's only one copy, if you only have bad copies of a lethal mutant allele, then it would be dead. Okay, so the only way that we can do complementation tests for lethal mutant alleles, recessive lethal mutant alleles, is by crossing carriers of the allele, heterozygotes for this allele, rather than homozygous lethal alleles. Okay, and the result's going to be interpreted a little bit differently. We basically have to look at the offspring and see how many of them survive, what percentage of them survive, right? So for something like fruit flies, you would kind of gather up all the eggs and see how many of those eggs actually survive to adulthood. In mammals, you might look at, for the number of pregnancies, how many of those survive birth and growth to sexual maturity. So different ways to look at it, but we're basically looking at percent survival of offspring when we cross carriers of different lethal alleles. So you discovered a lethal mutant allele, and you're going to cross it with the lethal mutant allele that someone else discovered to see if there is complementation with a slightly modified version of a complementation test. So the first modification is we're going to cross carriers of the lethal allele instead of pure breeding strains of the lethal allele, just because we can't make pure breeding strains. So the next modification we're going to make to our complementation test so that we can study lethal mutations with them is how we interpret the results. All right. So if we see that only 75% of the offspring survive to adulthood, then we would say that there's a failure to complement. Whereas if all of the eggs survive to adulthood, then we would conclude that complementation occurred. Okay, so instead of complementation being represented by all wild type offspring, as we saw in normal complementation tests, we would look for all 100% survival of offspring as a sign of, as our evidence of complementation for recessive lethal mutations. Whereas if only 75% of the offspring survive to adulthood, then we would count that as failure to complement. Okay, so here's why failure to complement would produce only 75% survival or 25% lethality. So if it's failure to complement, we're basically saying that the lethal alleles that we discovered and that the other strain has that we're comparing with, that we're doing the complementation test with, we're basically saying that these lethal alleles are mutant versions of the same gene. All right. So if we were to look at the Punnett square for this, we'd be looking at, notice that both parents are providing alleles for the same gene, the I gene, right? And notice that they're both heterozygous for this gene and that they have one wild type copies just so that they can be alive and survive long enough to reproduce. But the other allele is a recessive lethal allele. But also notice that the recessive lethal allele from each parent is different. This is the one we discovered. This is the one that someone else discovered. Okay, so if we were to then look at the results of that cross, right, the homozygous wild type one will definitely survive. Since the wild type allele is dominant for survival, then these would survive and these would survive as well. But 25% of the offspring will fail to survive to adulthood because they have two messed up lethal mutant alleles for this gene. They do not have the essential for life and development kind of wild type allele to help us survive. So these individuals are going to die, right? So that kind of explains, so since these ones don't have the working wild type version of this gene, they don't survive. So you see overall from this Punnett square that when you have kind of two mutant strains that are heterozygous for a recessive lethal allele, that 25% of the offspring will die, 75% of them will survive to adulthood. Okay? So for the other kind of situation is if we see complementation, if we see that 100% of the eggs actually survive to adulthood, we'd conclude that there's complementation. So let's look at what that would look like in terms of the genotypes. So let's say that for our mutant strain, right, it is a carrier for a particular mutant kind of recessive lethal allele. We'll call that A, right? So in terms of genotype, it's heterozygous for the A gene. It's got one good copy to keep it alive and help it survive, but it's carrying one copy of the recessive lethal allele, and it's homozygous wild type for this other gene. The other strain that we're comparing with, that we're doing complementation test with, it is heterozygous for a different recessive lethal allele for a completely different gene, the B gene instead, all right? And it's wild type for the A gene. So when you cross these two mutant strains together, which are carriers for mutant alleles of different genes, the A gene versus the B gene, then the offspring is always going to have at least one wild type allele for each gene, right? And therefore be able to survive. So for example, for the A gene, the offspring are always going to get a wild type copy from this parent, and it could be wild type or mutant for the other, right? But overall, they'll survive because it has a wild type A gene. Also, for the B gene, it always gets a wild type version from our strain, and therefore it survives as well. Okay, so in this case, we're basically seeing that each parent's lethal allele, so this lethal one here, is complemented by the homozygous wild type allele for the other strain. This mutant allele is being complemented by the homozygous wild type alleles in the other strains, in the other parent or in the other strain. Okay, so this slide here basically walks you through how we do a complementation test for recessive lethal alleles and how we interpret the results, which is a little different from how we do regular complementation tests. Okay, if you have any questions about that, do feel free to drop by office hours or just give me a call anytime during the weekday. So next, let's look at how we might organize some different new mutations into complementation groups. You'll see what this means in a minute. So let's say that you want to study all of the genes that might be involved in fruit fly making wings. So a way that you could start is by requesting all the different wingless strains that you can find from all the different labs across the world. So you read up on all the papers where someone has found a wingless mutant strain and ask them to send you some live samples. And in order to get even more wingless strains, you could take your wild type fruit fly colony and expose it to radiation just to produce more mutations and find more to create more wingless mutant strains. So let's say that from all that work, you collected 10 different mutant strains. So we'll call them strain W1 to W10. So wingless 1 to wingless 2, wingless 3, wingless 4, and so forth. So these are all pure breeding populations of fruit flies that have a recessive allele that's homozygous for a recessive allele that causes winglessness. But among those 10 mutant strains, it could be that you have affected the same mutant gene 10 different ways and 10 different times. So you just have 10 different mutant alleles for just one of the genes that's needed to make wings. Or conversely, it could be that for these 10 mutant alleles, they affect 10 completely different genes, and all 10 of those happen to be essential for making wings, which is why when you're homozygous for the mutant allele for any one of these 10 genes, you end up having a wingless phenotype. Or it could be something in between. So maybe you've identified 3 or 4 kind of different genes, and among your 10 strains, there's a few different mutations among those 3 or 4 different genes. And this is most likely that you're something like that, something in between, that you found more than one gene, but you have multiple alleles for some of them, right? So how do you figure out which situation isn't exactly how many genes you have affected and how many mutant alleles you've found? Well, basically, you just have to do complementation tests, but you do that for every possible pairing of these 10 genes. Now, that's going to be a lot of different complementation tests, so it helps to have something like a chart, a table like this, a matrix to help you organize not just the crosses. So you have here, I'm going to take strain 1 and cross it with strain 2, 3, 4, 5, 6, all the way to 10, and to take strain 2 and cross it with 3, 4, 5, 6, and so forth. It also helps us to organize the results of those tests, which are these pluses and minuses here. Right, so here are the 10 strains this way, 10 strains this way, and then just like a Punnett square, we're seeing what happens when we kind of cross these two to see what this becomes. And to help you understand the results, here's a legend. So if you see a plus in this complementation matrix, that means that complementation was observed when we crossed, in this case, strain wingless 1 with the wingless strain 3, meaning that when we crossed those two pure breeding strains together, we saw that all the offspring had normal wings. So they show complementation, meaning that 2 different genes are affected between the one that causes winglessness in strain 1 and the one that causes winglessness in strain 3. If we see a minus, it means that the 2 strains failed to complement, meaning that when they looked at the offspring, they still had no wings, they still had a mutant phenotype, which means that the gene that was affected in strain 1 and the gene that's affected in strain 2 are the same gene. Okay, so notice that this analysis, as much as it took a lot of work, it doesn't actually tell us anything about what the genes themselves do. We just know that they're necessary for making wings, but we don't really know anything about kind of exactly what they do in order to make wings. The point of this complementation matrix and all the complementation tests is just to do the early stages of, now that we have many genes important for making wings, figuring out many strains that are mutated and unable to make wings. We just want to know how many different important wing-making genes have we identified among these 10 strains? All right, so let's look at how we would interpret these results. So let's look at it one row at a time, starting at the top. So we're going to look at the row where we're taking strain wingless 1 and crossing it with all of the other 10 strains. And notice that every time we see a minus, that tells us that when strain 1 was mated with strain 2, it failed to complement, and that tells us that the gene that was mutated in strain 1 is the same gene as the one that's mutated in strain 2, which is why they failed to complement. Okay, so that means we can put strains 1, 2, and 7 into the same complementation group. So let's call that group 1. So group 1 is the wingless strain 1, 2, and 7. We do this because they fail to complement, and also it tells us that the alleles we find, the mutant alleles we find in strain 1, 2, and 7, they're all alleles of the same gene. So we can call that gene 1 if we want. Okay. Now we can actually technically skip row 2. As much as we're going from top to bottom, we can skip row 2 because we've already assigned strain 2 to a group. So we're not going to get any new information here. But just for practice, let's look at what would happen if you forgot to skip row 2 and you try to take a look anyways. So notice that if you looked at row 2, you'd find that, okay, so row 2 has a minus. Obviously, it fails to complement with itself because it's going to be the same gene affected in strain 2 and strain 2. If we continue on, there's also strain 7 also fails to complement with strain 2. So we'll just see if we look at row 2, once again, that strains 2 and 7 should be grouped together, but we already knew that, right? We knew from row 1 that 1, 2, and 7 belong together. And the reason why we start at the top with the most tests being done is that we end up grouping all of the strains that belong together when we take the larger rows first. Okay, so there's really no need to look at any rows for the strains for which we've already identified a complementation group. Okay, so we're going to skip row 2 and move on to row 3. So for row 3, we're going to group together all the strains, once again, that have a minus. So basically fail to complement as shown by the minus sign here. So that would put strains 3, 4, 5, 8, and 9 into one complementation group. So we'll call that complementation group 2. Once again, because they each show a failure to complement with strain 3, and therefore they must all be a mutant because of a mutant allele found on the same gene. Okay, so again, since we've already grouped, so the next row is going to be strain 4, which we already grouped, row 5, which we already grouped. So row 6, we're going to skip row 3, 4, and 5 since we already put that in group 2. Move on to row 6, and here we see that strain 6 and strain 10 fail to complement, so we can put those two into the same complementation group as well. So that's group 3, and then we can stop. We can stop because if you count up the number of strains we've already assigned to groups, there's 10 there. We've assigned all 10 mutant strains. So having done that, here's a kind of participation question for you to think about, right? So in terms of genes that have been affected by mutations among these 10 mutant strains, how many genes are represented here? All right, so pause the video and make your choice. All right, welcome back. So correct answer is C, right? So we have 3 different complementation groups, right? So one group, so these 3 mutant strains affected this particular gene. These 5 mutations affected gene number 2, and these 2 strains are mutated, have mutations that affect a 3rd gene. So 3 different genes are affected. Okay. Now next, let's focus in on the gene that's represented by group 2, right? The one that is mutated in strains 3, 4, 5, 8, and 9, right? How many different alleles do you have among these 10 mutant strains? Okay, assuming that when 2 mutant strains are isolated and created independently, that the version of the mutation, the type of mutation, the allele that you get is different. Okay, it's not the same identical mutation. Okay, so pause the video, make your choice. All right, welcome back. So the answer is D, right? So a lot of you would have gotten this wrong. Most of you probably chose C, saying that there's 5 mutant alleles. So let's walk you through why it's 6. So we see that group 2 has 5 different mutant strains, and since we isolated these 5 mutant strains independently from different labs around the world or created them brand new in our lab, like the likelihood that they have the exact same genetic sequence, the exact same allele, is very, very low. So we can assume that these 5 mutant strains each are homozygous for their own unique mutant allele. So that's 5 different mutant alleles. But there must also be a wild type allele around, right? So for example, when you take strain wingless 3 and you mate it with strain wingless 6, the reason why all of the offspring end up having normal wings is because wingless 6, it has a wild type allele for gene number 2, for the gene that's affected here, which is why I can provide what these strains are missing and make wings for these offspring. Strain wingless 6 just has a mutation in a completely different gene, for which strain 3 has the wild type allele. Right? So basically, in order to have complementation, the other strains, the ones that are not in group 2, must be homozygous for the wild type allele. So since the question asks you how many different alleles do we have for gene 2 among all 10 strains here, then we would count up the 5 mutant alleles found in these 5 different mutant strains in group 2, but also keep in mind that there is a wild type allele in all the other strains that are not part of this complementation group, which is why they're able to complement with these strains. Okay, so some people get confused by that, so do call me or drop by our office hours if that's confusing. We can discuss this some more. Okay, so at this point, we've confirmed that we have among these 10 mutant strains 3 different genes that seem to be necessary for making wings, and we actually have anywhere from like 2 mutant versions of that mutant alleles for that gene up to like 5 different mutant alleles for one of these important for making wings kind of genes. Okay, so these are genes that are essential for making fruit fly wings. All right, so that's kind of some of the work that you can do as a geneticist to help understand how important biochemical or developmental processes work based on doing complementation tests, just to sort through the strains you have and to see how many genes you've identified or have isolated mutations for. Okay. All right, so next, let's take a look at how we might study biosynthetic pathways. So this example kind of looks at the beginnings of the work you might do if you're studying the developmental pathway for making wings. But we can also look at biosynthetic pathways, meaning pathways which are a bunch of steps necessary for making something that's biologically important. Okay, so synthesizing something. So a lot of useful substances such as, the amino acid arginine, which is one of the amino acids you need for making a lot of different proteins. A lot of funguses and microorganisms and animals and plants as well can make their own arginine, but it does so through a series of chemical reactions. It's not like you can just take sugar and turn it into arginine with a single step. Instead, you take some sort of early precursor that you may have absorbed from digested food, and through chemical reaction after chemical reaction, you change it from one thing to the next until finally the final reaction creates arginine, basically producing the amino acid that you'll need for making other proteins. So for each step of this biosynthetic pathway, for every time that you change the chemical from one chemical to another chemical to another chemical, you need an enzyme to catalyze each of these reactions, a unique enzyme. Now, each of these enzymes presumably has one or more genes that are needed to produce that enzyme. Okay, so there's a lot of things that you can study and figure out that have to do with the production of arginine. For example, we need to figure out what these enzymes are. We need to figure out what genes are responsible for making those enzymes. We also want to know what are these intermediate chemicals and what's the starting chemical, the precursor we need in order to make arginine, right? So to fully understand this biosynthetic pathway, we need to figure out all of this information. So how do we start? How do we even begin this research? Well, similar to what we did with the wingless flies, we start with mutant strains that can't do this successfully, for which some part of this process is broken. Okay, so we need to make some mutant strains that are not able to make arginine, right? Then we can use some experiments to figure out where is this biosynthetic pathway broken, and from that figure out what the green words here are, what are the intermediate chemicals, what are the enzymes, what are the genes. So we can figure out all these things, starting with strains that are unable to make arginine. Okay, so we can figure out the precursors, the genes, the enzymes, and kind of understand the chemical reaction thoroughly using these kinds of experiments that researchers named Beadle and Tatum did this, actually back in the 1940s. Okay, so let's walk through how these researchers did this step by step, and they used Neurospora, which is a filamentous fungus, as a model. Okay, so The fungus is the fungus is a eukaryotic organism that you can easily grow in the lab on agar media, which is kind of like a solid jello-like media that provides the nutrition needed for things to grow. Okay, so let's start. Say that we started with a wild-type prototrophic fungal strain. So for this particular species, we picked the wild-type, the normal, found in the wild fungal strain that happens to be prototrophic, meaning that prototrophic means that it's able to synthesize every amino acid and vitamin that it needs for itself. Okay, which basically means that if you just provide it with some sort of energy and carbon source, like glucose as a sugar and a bunch of inorganic salts, like nitrogen and phosphorus, the kind of things you'd find in fertilizer and in the soil, then the fungus doesn't need you to give it any amino acids or vitamins. It can make its own from those basic building blocks and that energy. Okay, so let's say that we then exposed this, because so as a starting point in our lab, all we have is this this prototrophic wild type fungus. So in order to create some mutations to study, we're going to take that wild type strain and expose some of its spores to X-rays, which is radiation that's going to damage the DNA and produce new random mutations in its genome. And then we're going to take those X-rayed, DNA damaged and mutated spores and grow those new individual strains on separate tubes containing complete media. Complete media is basically agar media, so a jello-like media that contains not just glucose and some inorganic salts, but also contains all the vitamins and amino acids that the fungus might need to survive. Right. So this way, even if the mutant strain is no longer able to make some amino acid vitamins for itself, we can still grow it because we need to let some of each of this fungus grow so we have more material to work with. Okay, so from here, what we'll do is we'll take a little bit of each of these newly mutated fungal strains and transfer it into a tube of a different type of media. Notice that media is represented by a different color here, because this media is now minimal media, which means that it doesn't have any amino acids or vitamins included. All it has is those inorganic salts and chemicals, plus some glucose as a sugar and energy source. Okay, so notice that for most of these tubes here that we kind of X-rayed, grew in complete media, and then transferred to a much less nutritious media, the minimal media, it can still grow, right? So whatever mutations might have happened or not, it didn't seem to have knocked out its ability to make anything important, such that when you put it into minimal media, most of them still grow, except for this one tube here, right? So now we're really interested in this particular strain that we've created, right? So what does it mean when this strain here can grow in complete media, but can't grow in minimal media? Right? So you can pause the video and think about that for a sec. All right, welcome back. This basically tells us that this strain here is an auxotroph. Okay, so a prototroph is a strain that can make everything for itself. An auxotroph is basically a nutritional mutant. It's mutated one of the genes necessary to synthesize some sort of essential nutrient. So if you deprive it of supplemental nutrients, you put it in minimal media with no organic nutrients provided, no amino acids or vitamins provided, it cannot grow. Right? You have to give it help, give it more nutrition to grow. Just like we as humans, we're kind of auxotrophs as well, in that we need our B vitamins, which are amino acids that we can't make for ourselves in order to survive. Okay, so notice what we did was we took a wild type prototroph that normally in the wild can make everything for itself, and by exposing it to X-rays, we've managed to create one here that is an auxotroph. It's lost the ability to make a particular nutrient that it used to have. Okay, so now we're really interested in this particular strain. So we're going to take some of this strain here that we've now confirmed as an auxotroph because it can't grow in minimal media. So we're not going to take it from this tube. We're going to go back to our original tube in complete media where it can actually grow, and take some of that fungus and put it into minimal media again, but this time we're going to supplement that minimal media with one of two things. We're either going to add back in all of the amino acids, so all 20 amino acids, or we're going to add back in all of the vitamins that I might need. Okay, so notice that when we did that, it manages to grow in minimal media when you add all the minimal amino acids back in, but not in the minimal media that you only added the vitamins into. Okay, so what does that mean? What does it mean that it can grow in this tube and not in this tube? Okay, so pause the video and think about what your answer might be. Okay, welcome back. So it basically tells you that whatever mutation was caused by these X-rays, it basically shut down its ability to make at least one important amino acid. Okay, that's why when you add all the amino acids back in, then it says, oh great, you've given me what I can't make for myself, I can grow again. But in this tube that only has minimal media plus vitamins, it's missing some amino acid. It doesn't have a supplement of amino acids to help it to grow. Okay, so it seems like we're on the right track since we're trying to find strains that cannot make arginine as a specific amino acid. This one has potential. What we found, we've created a mutant strain that is unable to grow unless you give it some amino acids. Okay, so to figure out exactly which amino acid this oxytrophic strain can no longer make, what we're going to do is take some of this strain and put it into minimal media, but this time instead of giving the full 20 amino acids that it might need, we're just going to give it one amino acid at a time. So we got 20 different tubes, each one containing just one amino acid. All right, and we're going to see which amino acid, when you add it back in, allows the strain to grow. And what we're looking for is this. We're looking for a strain that grows when you add arginine to the minimal media, but not when you add anything else. So now we've found an arginine oxytroph. Okay, we found the newly created oxytrophic fungal strain that cannot grow unless you give it arginine through its growth media, because it cannot make it for itself anymore. Okay, so this is the strain that we really want to study because we want to understand what did we break, right? What gene was originally necessary to make its own arginine? All right, so let's say that we repeated these steps many, many times and was finally, after a year or so, lucky enough to isolate 3 different independently created arginine oxytrophic strains. Right, so we'll name them arginine oxytroph 1, 2, and 3. So strain 1, 2, and 3. So what would be the next step? What should you do next? Well, first step would be to do complementation tests again, right? We have 3 different mutant strains that cannot make arginine, but we don't know if these 3 different strains have mutations in the same gene or different genes. So let's say that we conduct a series of complementation tests, right? So we did every possible combination of pairing of arginine 1 and 2, 1 and 3, 2 and 3, and so forth. And we found complementation for every pairing that we did. So what does this mean? How would you interpret those results? So pause the video and make your choice. Okay, so welcome back. So the correct answer is B. So if we see complementation when we do complementation tests between all the different possible pairings of these 3 mutant strains, that means that we're seeing the wild type phenotype for the results of that cross, and that tells us that different genes are affected in each of these 3 mutant strains. All right, which is great because that's what we wanted. We wanted to kind of isolate and identify different genes involved in making arginine so that we can better figure out what genes, enzymes, and intermediates are involved. All right, so we made some good progress with our research so far. So as a next step, we're going to take each of these 3 oxytrophic strains and grow them in minimal media. And this time we won't give it, we will give it arginine as a control, but instead of giving it just arginine or any other amino acid, we're going to put in chemicals that are intermediate chemicals that are in the biosynthetic pathway, partial steps towards producing arginine. And in doing so, we'll be able to try to figure out what the biochemical pathway is. So what is the identity of these 2 intermediates leading up to the production of arginine? So here are the results of that experiment. So we basically have minimal media that's supplemented with either arginine itself or 2 different intermediate chemicals. And we see when we put each of these mutant fungal strains into these tubes of minimal media plus just one of these supplements at a time, we can see whether plus that strain is able to grow or minus that strain is unable to grow. So considering that kind of quick explanation of how to read this table, what would you say is the identity of intermediate 1 and 2 in this biosynthetic pathway for synthesizing arginine? All right, so here are your possible answers. So pause the video and make your choice now. All right, welcome back. So the correct answer is A, and it takes a little bit of thinking to figure this one out. So if you didn't get this answer correct, that's totally fine. I just wanted you to try to logic it out. So this is how you can kind of think your way through it. So I'm just going to go ahead and put ornithine in here for intermediate 1 and citrulline here for intermediate 2, just to kind of complete the pathway because it makes it easier to discuss afterwards. All right. So, one of the ways you can approach this question, if you read it and you kind of forgot how to interpret these, is kind of look at the end result, right? So, notice that by adding arginine, which is kind of the latest step, the last step in this biosynthetic pathway, we're obviously able to help each of these arginine mutants recover. So, logically speaking, it makes sense that we isolated these arginine mutant strains um, by by testing them to see that adding arginine is what they need in order to keep growing. So obviously adding arginine back together helps, but it also helps you to orient yourself between this data and the biosynthetic pathway, knowing that the later you are in the biosynthetic pathway, then the more strains you're able to rescue. Okay, so therefore, citrulline, the chemical that can rescue the second most number of mutant strains, that must be fairly close to arginine because that's the most similar result to arginine, and the one that's least similar to arginine must be further away in the biosynthetic pathway. So that's one way to interpret these results in a way, just by orienting yourself based on this kind of easy endpoint here. All right, another way to look at it is in terms of why a particular supplement can support the growth of more mutant strains. So the later a supplement appears or an intermediate appears in the biosynthetic pathway, the more mutant strains it can rescue. Because if you think about it, whether it is this enzyme or this enzyme that's not working, whether it's the gene making this enzyme that's mutated or the gene making this enzyme that's mutated, right? If you add a chemical that comes later in the biosynthetic pathway, all it needs is this one last enzyme to work to make arginine, and that's going to rescue the strain. It's going to allow the strain to grow. Okay, so that's what citrulline is. Citrulline is late in the biosynthetic pathway. Therefore, as long as enzyme C is present, as long as the gene for making enzyme C is present, you're going to get arginine. Doesn't matter if either or both of these are broken. Okay, whereas if you have a chemical like ornithine, which appears earlier in the biosynthetic pathway, right? Even if enzyme A is working, if enzyme B or C is broken, right, as we see in arginine 2 and 3, then we're not going to be able to make arginine, right? So the earlier the supplement is, the more genes and enzymes have to be, the more wild type alleles have to be there, the more working enzymes need to be there for this pathway to finish from there onwards to make arginine. So that's another way to look at it. All right. So different ways to interpret these answers, whichever works best for you, whichever logic that makes most sense to you, make sure feel free to use that method. Okay, so here's just some quick examples. So if, for example, enzyme B is what's kind of if the gene for making enzyme B is homozygous mutant in a particular strain, then adding citrulline will allow it to make arginine and survive. By adding ornithine, it won't, because you don't have the enzyme for change ornithine to citrulline, and therefore you don't have any citrulline to make an arginine. Therefore, you don't have arginine, and it doesn't survive. Okay, so we've made some really good progress in our goal of figuring out all the details of this biosynthetic pathway, right? So we identified these, we basically created and isolated these 3 mutant strains, which affect 3 different genes. And by adding these different supplements and looking at our results, we figured out the order in which these chemicals appear in the biosynthetic pathway. So using these same results, we can actually also determine which enzyme's gene is mutated in each auxotroph. Okay, so to help you understand the question better, so for each of these mutant strains, they have just one gene that they're homozygous mutant for, and therefore there's only one enzyme that it's unable to make. So I'm asking you, in terms of genes, for each of these strains, which enzyme is that strain unable to make because it's homozygous mutant for that gene? All right, so with that in mind, pause the video and make your choice. All right, welcome back. So the correct answer is A. So notice that mutants that have a defective enzyme earlier in the pathway, right, they tend to be able to grow with a wider variety of supplements. All right, so for example, arginine 1, we concluded from our answer here that the gene that's missing is the one needed for making that. So the wild type allele that's missing is for the gene that's needed for making a working enzyme A. All right. So if the only part of this biosynthetic pathway that's broken for this mutant strain is the first enzyme, then that's kind of a good thing because you can add any of the remaining chemicals further down the line, and all the enzymes you need to make arginine are there, right? You can add ornithine, and you have enzyme B and C and so forth. So that rescues it. Citrulline works as well because there's enzyme C to make arginine, or if you add arginine itself, of course, it survives, right? So that's why you see the most pluses when the broken enzyme is earlier in the pathway. Okay, so later enzymes are still working. So the opposite is also true, right? If you find that a for a particular mutant strain, it is a later enzyme that is mutated. So for arginine 3, this mutant strain, it is homozygous mutant for the gene needed to make enzyme C. So if this is broken, then you can add anything before this, is not going to work because the very last step cannot be done without enzyme C. So the only thing that can rescue it is if you add the final result, arginine, instead. Okay, so that's kind of like the two extremes that you can look for. So generally speaking, when the gene for an enzyme is mutated, right, then only the chemicals that appear downstream of that particular enzyme's kind of reaction will be able to support the growth of that auxotroph. Okay, so kind of explaining these situations in 2 or 3 different ways. Hopefully you'll find one that works well for you. If you're still confused about this, do feel free to drop by my office hours or give me a call. I'd be happy to explain it further. Okay, so we've done a bunch of work and we're kind of done here, right? So by identifying these strains and doing this experiment, we're able to first figure out the order, the identity and order of these different intermediate chemicals, and then figure out exactly which which gene affects which enzyme, or which mutant strain is deficient in which enzyme. Okay. So what's the point of doing all this work? I mean, that took a lot of work, especially in the early stages, to make and isolate those particular mutant strains. So what's the point of deciphering these biochemical pathways? Well, since a lot of you guys are interested in med school, an interesting medical example is phenylketonuria. So it used to be a lethal recessive disorder. So it's a disorder caused by being homozygous recessive for a particular gene. And it used to be lethal, meaning that the individuals who are homozygous recessive wouldn't survive past being a baby, essentially. But it's no longer lethal because we know how to treat it, and it's a very simple treatment. So this is what happened. So we discovered that the homozygous recessive individuals, just like for the arginine mutant fungal strains, because they're homozygous mutant, they're unable to make a particular enzyme, and that enzyme is needed for a particular reaction. In this case, it's needed for a reaction that helps us take phenylalanine and convert it into tyrosine, a different amino acid, as well as a bunch of other useful chemicals. So phenylalanine is an amino acid you would get and absorb into your bloodstream when you're eating proteins. And normally you would have a working enzyme to change that into other useful chemicals. But those who have phenylketonuria, those who are homozygous recessive and do not have a working copy of this gene needed to make this enzyme, it can no longer do this reaction, right? So that's a broken step in this biochemical pathway. So what happens instead is that phenylalanine ends up building up inside the bloodstream and inside the body, and to deal with that, the cells in the body basically convert that into something else, phenylpyruvic acid instead, and that phenylpyruvic acid is very toxic to the development of brains, which is very important in babies, which are very actively developing their brains, right? So as a result of this buildup of phenylpyruvic acid, because of this kind of blockage, this lack of ability to catalyze this reaction, that causes neurodegeneration and death in babies, which is why it's a lethal disorder that acts early in life. But because we now understand the biochemical pathways that got broken and also the alternative pathway that gets taken that becomes toxic to the baby's brain, we can actually treat this disorder just by avoiding the buildup of phenylalanine to begin with. So the main treatment for this is to avoid high protein foods, to decrease the protein intake, particularly in a baby. So for a baby that's still developing its brain, you would kind of very carefully control the protein intake from these for these infants so that they don't build up these toxic chemicals to high enough levels to basically kill the brain and kill the baby. But also you would limit protein intake in a mother who is affected by this disorder, because she also doesn't want to build up phenylpyruvic acid to high enough levels to not poison her because her brain's already fully developed, but to poison her fetus, because those toxic chemicals in her blood will also be passed on to the fetus as well. Okay, so once you've helped this affected individual to survive to adulthood, we also have to protect her offspring as well, in this way. So a very simple treatment of just minimizing protein intake, but that made it what was previously a fatal disease into someone that someone can not just survive but reproduce while being affected by this disorder. Right, so that's kind of a cool example of how all this research into genes, gene interactions, and biosynthetic pathways can be useful for preserving human life and creating human life, actually. So pretty cool stuff. So that's it for this lecture. If you have any questions about it, please do feel free to drop by my office hours or to give me a call, and I'd be happy to explain things further. But I'll just leave you now with another random video of my kids doing stuff. All right, so enjoy, have a great day, and hopefully I'll talk to you again soon. Bye-bye.